Hello, my lovely anatomists and physiologists. Michele Glass here, and we're still talking about vision. I wanted to talk a little bit about how those photoreceptor cells are stimulated. And we're going to focus in on the rod cell, which remember is our low light photoreceptor. It's often described as like black and white vision. It has the opsin molecule called rhodopsin. And so rhodopsin here, in this case, is our visual pigment. And a pigment, remember, is a molecule that's going to absorb wavelengths of light. Now, in the case of our rhodopsin, this is going to be a transmembrane protein in those membranous discs of that rod cell. And it's going to be composed of both what's called opsin and then retinol. Retinol is related to vitamin A, and it's going to change shape when it's hit by a photon. So it's going to change from a cis shape or a bent shape to a trans shape or a straight shape when it's hit by light. So it often is depicted in our diagrams as kind of this maybe square shaped structure with a line that's then like bent. So I kind of think of it as looking like a key a little bit, but it's got like a bent key. And then when it's in its trans configuration, then it's just completely linear. There's no bend to it. So it's looking a little bit more like a key. And so what we see is when photons hit retinol, it changes from the bent shape to the straight shape. When it's in the straight shape, it can't be simulated again. So we go through a process where we have to bend it back. When we're talking about cis and trans in this context, this is coming from our organic chemistry discussion. And it's just looking at the organization of the molecules, or the atoms in the molecule, I should say. So what we're going to see happening essentially is our opsin and our retinol are together as a molecule. And we start out with our retinol in this cis shape, which I just described as being bent. And then a photon hits it and it straightens up, right? And when it straightens up, we can say, okay, two things are happening here. Number one, this sends our signal on to increase sodium permeability of the rod cell. So y'all should know this: if sodium is flooding into the rod cell is depolarized. And so that's sending that signal onto the bipolar cell, which sends the signal on to the retinol ganglion cell, right? And then that's becoming the signal that travels through the optic nerve, cranial nerve number 2. But then we still have to deal with, oh my goodness, our retinol is trans and it needs to be in the cis shape. Our retinol is straight and it needs to be in the cis shape. And so we talk about this as bleaching, because right here we can't see anything. We can't stimulate this molecule again. So basically what we see happening is the retinol and the opsin are going to separate, or we can say dissociate, okay? And then we're going to see ATP is going to be used to change the shape from the trans shape back to the cis shape. So we're going back from the sort of key, straight key, to the bent key. And once we have that bent key, now we see that the retinol is, let's say, reincorporated to the opsin and ready. I'm just going to say ready, which basically means, okay, we're back to our start and we can be stimulated again. If you've all ever experienced when somebody puts a flashlight in your face and it's a really bright light, or you look up at the sun, you walk from the dark movie theater to the sunny outside, any of these scenarios where really quickly you have photoreceptors that are being stimulated, and then you have kind of a momentary sort of blindness where you can't see anything, right? You've just bleached a whole bunch of those photoreceptors, right? You've just stimulated them all, and you have this momentary period of time where you have to have the retinol and the opsin separate. You have to get an ATP to change the shape of the retinol back to the cis shape, and then you have to reincorporate it before it can be stimulated again. You know, your rod cells in really sunny conditions are constantly being bleached. And so that's where your cone cells come into play to help you and highlight scenarios. Okay, now that we understand what's happening with the photoreceptor, now we're ready to work through our afferent pathway. So stay tuned for that. Up next, as always, take care of yourselves and each other.